Hi, this is Mrs. Sloan, and this is a video intended for my honors biology class, and this is about cell structures and functions, and this is part one. Here in part one, we're going to talk about the cell theory and why cells are small, and then we're going to talk about prokaryotic cells. And then in video two, I will talk about the structures of eukaryotic cells. All right, so down in the descriptor of this video are the group shared notes. And if you would like to have a copy of those, you can open those up. And I present in two columns. Let me move myself here. I present in two columns. Column one is the scaffolding of the notes, and then as I'm talking, my students fill those in. And then column two, you should throw in pictures or things that can help you review the material. Maybe something from this video, you can just pause it and get a screenshot and throw it right in there. Okay. All right, let me move that. All right, so this is about cell structures and function. So the first thing I want to go over with you is if you take a look at this picture, there's actually multiple cells on here, and I want to introduce you to the difference between a prokaryotic and a eukaryotic cell. What I'm outlining right now, okay, this big outline right here, that is a eukaryotic cell, and probably that's the nucleus of the eukaryotic cell, the brain. So that's how large a eukaryotic cell is. What you're looking at here are hundreds or thousands of little E. coli. Those are the dots all over those. Those are prokaryotic cells. They are much smaller. If I was going to do a contrast between these two, think of a prokaryotic cell like a one-room apartment. Literally, the kitchen sink and the stove and the bedroom, everything is in that one room. That would be a prokaryotic cell. And then a eukaryotic cell would be a ginormous mansion full of multiple rooms, and each room has its own specific function. All right? Now, regardless if they're big or small, okay, we need to go over the cell theory. Remember, this is a theory, right? So, number one, all living things are composed of cells. If it's alive, it's made out of a cell. It might be one cell, it might be trillions of cells, but it is made out of cells, and that is the structural and functional unit. That's how that living organism works. Remember, for my students, we have already talked about atoms. Atoms build molecules. Molecules can build organelles in eukaryotic cells, and then organelles cells, right? So that living thing, remember, it starts at cells. So cells build tissues, organs, organ systems in an organism, all right? And then the third is that you're not going to get any more cells, and all your cells have to come from pre-existing cells, okay? So on your notes for the cell theory, all living things are composed of cells. The cell is the structural, here, I'll back it up so you can see it. The cells are the structural and functional unit of organisms. And then number three is cells come only from pre-existing cells. All right, cells are very small, some cells smaller than others. This cell right here, these two cells right here are eukaryotic cells. Those are your larger cells. This is a typical animal cell and a typical plant cell. Now, when I say typical, if I said typical student, are there any really typical students? No, you are all unique and different, and with different strengths, right? So this is just kind of a generalized, would probably be a better term for it, and a generalized plant cell. Now, I know you know what the powerhouse of the cell is, mitochondria, right? This mitochondria might be about the size of a prokaryotic cell that you would see. All right, and we're going to be learning about all the parts of these eukaryotic cell organelles in video two. All right, now, why are cells small? Okay, well, first of all, what we need to know is you want to maximize the surface area to volume. So that's why cells are small, and I'll be talking about that in another slide. But you can see again, contrasting, a prokaryotic cell is maybe 1 to 10 micrometers. Now, let me tell you, there are 1 million micrometers in a millimeter. So if that gives you some, scale there, okay? So there are 1 to 10 micrometers in a prokaryote. That's the standard size of a prokaryotic cell. And then you could be up to 10 times that in a eukaryotic cell. So you can see that right here. plant and animal cells, you can see a chloroplast, which is an organelle for a plant cell. Look at where most bacteria fall, okay? Obviously, this isn't a super good scale. Otherwise, mice would be terrifying and whales would be pets. Let's see, what else do I need to get here? No, I think we're good. Okay, next, if you wanted to calculate surface area to volume, what you would do to calculate surface area is height times width times number of sides. And if you're talking about multiple cubes, then you would multiply it by cubes. That will give you the total surface area. And then volume is height times width times length times number of cubes. So you can do the math on that. Now, I want you to see, you can see that 1 4 centimeter cube, if you did the math on that, okay, then if you calculate the surface area, so that would be height times width, right, which would be 4 times 4 is 16, times the number of sides on a cube is 6, and it's 1 cube, and you divided that, right, and if you went by the volume and you did height times width times length, okay, that would be 4 times 4 times 4 is 16 times the number of cubes. Then your ratio of surface area, total surface area to the volume is 1.5 to 1. Now, if we take that same volume, but instead divide it up into 8 cubes, you're increasing the surface area, right? Because we don't just have these 6 edges, right? You have the 6 edges of this, and then 6 times 8, you've increased the number of sides, right? So you're increasing the number of sides, and as a result of that, your surface area grows per your volume. Right? And then if you go up here to 64 cubes, you're increasing the surface area even more for that same volume. Remember, the edges of the cell is where everything's going to go in and out of that cell. And so you want to maximize that. Also, whatever goes in that cell, you want to make sure it gets to all and permeates all areas of that cell so that the cell can complete whatever function it wants to do. And I'm going to give you a quick analogy of that, okay? I remember when the cologne Axe was super popular. I never really liked the smell of it. But anyway, some guys put a lot of Axe on their body. And you're like, ugh. Okay, now, let's imagine somebody who has oversprayed Axe and they walk into 3 different rooms. They walk into the gym at school, they walk into my classroom, and they walk into an elevator. Now that Axe leaving their skin is going to travel at the same rate, whatever the temperature is of the skin, right? It's going to travel at the same rate, but which room will it permeate faster? Which room will it get from one side of the room to the other? The elevator, my classroom, or the gym? And hopefully you obviously see it would be the elevator, right? Because it has a shorter distance to travel than to permeate a whole room or an entire gym. And that way, whatever comes into that cell is permeating that cell, and that cell is being able to react to that in order to maintain homeostasis, all right? So this is why cells are small, increasing the surface area to volume, and so that something can travel through that cell, getting from one side to the other more rapidly, all right? So smaller cells have more surface area per volume than larger cells. This is why cells are small, okay? So on your notes, cell size, I already gave you cell size, 1 millimeter to 1 micrometer, somewhere in that range. The requirements, cells need a large surface area, plasma membrane, to adequately exchange materials. So the surface area to volume requires that cells be small. And then there can be modifications on the edges of the surface to increase the surface area, like folds would do that. All right? Good. All right, so now we're going to start talking about prokaryotic cells. So the first thing I want to talk to you about prokaryotic cells is look at all these different shapes you can have, right? Okay, but now let's look at this really simple prokaryotic cell. You're going to see there's no mitochondria in there, there's no chloroplast, nothing like that, because they have no membrane enclosed organelles. They do have everything they need, right, to survive and reproduce. Bacteria are the most numerous, right, cell probably on the planet. In fact, there are more bacteria on just one hand right now than there are people on the planet. That's how numerous bacteria are, right? And so when you look inside of it, this like blue mess that looks like something that a cat coughed up, that is a single circular chromosome that has it's been all kind of crinkled up, and that single circular chromosome, where you and I, we have linear chromosomes, but prokaryotic cells have that circular chromosome, and it's all fitting into this one region, and that's called the nucleoid region. Now, the fluid of the cell is technically called the cytosol. That's the fluid portion, or it's referred to as the cytoplasm. Now, within that, do you see how they have ribosomes? These ribosomes, what we're going to learn, this is the workbench for protein synthesis. So DNA is your code book, okay? And from that DNA, you transcribe that DNA into mRNA, and mRNA will go out and get on a ribosome and build proteins out there. So it's the way you enact your genetic code is proteins. That's how you enact your genetic code. So making proteins is really important. So you have a lot of ribosomes in here. Now let's look at these different layers that you can see. Now, what all cells have is for sure a cell membrane. Okay, that is the border of every single cell, but that will be the most interior border. Some cells have more than that. For instance, this bacterial cell, it has, and bacteria do, they have cell walls that are on the outside of that. Now, that cell wall is not the same kind of substance to build plant cell walls. Plant cell walls are built out of cellulose, a polysaccharide. They are built out of something called peptidoglycan. I'm not holding you to that. I'm just letting you know it's a different substance, just like fungal cell walls are built out of chitin. All right. Now, external to that cell wall is a layer basically of carbohydrates that have all these like little hairs. Those little hairs are not cilia. Prokaryotic cells don't have cilia. They don't use cilia for movement. What prokaryotic cells, if they're going to have something to help them move, is this long thing right here, and that's a flagella. Okay, so that flagella is plural, so technically that's flagella. And that is what's on the end of sperm in eukaryotic cells to help sperm swim, right, on the fallopian swim team. All right, so what you see here is this carbohydrate capsule on the outside, and these little things are called fimbriae. And fimbriae help them to grab onto surfaces like the linings of your intestines. All right? So what do they have in common? They all lack a nucleus, and they lack all other organelles as well. But they all lack a nucleus, but they do have the DNA. Number 2 on the notes, they are smaller and simpler than eukaryotic cells. Smaller and simpler than eukaryotic cells. And there are 2 domains that prokaryotic cells fall into. And we have reviewed this a little bit already, but I'm going to hit it again. It's bacteria, B-A-C-T-E-R-I-A, bacteria, and archaea are already on there. That word's already on there for you on your notes, A-R-C-H-A-E-A. All right, and we'll talk a little bit more about those again in a minute. So now I'm going to show you a little more complicated picture. Oh, no, I'm not. I lie. I am going to show you a picture of a phospholipid bilayer, and this is what makes up the cell membrane, okay? That membrane that goes around all cells. So here, remember when we learned about the 4 important organic molecules, right? Do you remember what they are? You should if you're in my class. Okay, carbohydrates, which are built out of sugars, right? Lipids, and there are 2 categories of lipids, fats and steroids. Remember, fats are made out of glycerol and 3 long fatty acid chains. And then the third one was steroids are made out of 4 rings. Proteins are built out of amino acids, and nucleic acids are built out of nucleotides. Good. All right. When we talked about modifying a fat, we said you could take the glycerol with the long fatty acid chains, and you could replace one of those fatty acid chains with a phosphate group. And that's exactly what this phospholipid is. The long fatty acid chains are hydrophobic, and you can see they're all clustering right here in the middle. And then these blue spheres are hydrophilic. This is referred to as being amphipathic when you have different regions that have different affinities for water. All right. So we can see phospholipids in our cell membrane. And then we can also see right here, this is a giant protein made out of amino acids. Now, we have a whole chapter dedicated to cell membranes, so I'm not going to go into much more detail than that, other than if you look at your notes, it says outside plasma membrane. You want to know this regulates the movement of substances into and out of the cell. It regulates the movement of substances into and out of the cell. Now, outside of the cell membrane, which is in blue, this blue color, I know this is a scary picture. Just hang on. It's all good. All right. Outside of this cell membrane in blue, right here is your cell wall. It's the next layer out. So it goes cell membrane and then it goes cell wall. And this cell wall protects, put this in your notes, protects the cell and maintains the cell shape, protects the cell and maintains the cell shape. In fact, when you take antibiotics, anti-life, because you have a bacterial infection, they don't work on viruses, they don't work on COVID, but when you have a bacterial infection, it can kill the bacteria in two different ways. One, it'll inhibit it from making any more cell wall for either repair, okay? Or two, if the cell replicates itself through binary fission, it cannot generate or build any cell wall, and that's how you kill off bacteria. Okay, so cell wall protects the cell and maintains the cell shape. Then you can have this carbohydrate capsule on the outside that you see in tan. It kind of looks like a corn dog that you dropped in a hair salon with like hairs all over it. But the corn dog outer covering right here, that is made out of carbohydrates, and it's referred to as a glycocalyx. And you can see right here, polysaccharides, which you should already be familiar with. Now, it can be like a capsule that's hard, or it can be slimy. All right. Now, attached to the bacteria, we've talked about one of those already, and that is the flagella, right? And that flagella is for movement. So you want to put on your notes, movement. And then all these things that look like little hairs, those are called fimbriae, and that is for surface attachment, okay, to help it stick to things. All right. Now, up here, little bit thicker than all these fimbriae out here is this sex pilus. Now, okay, so let me make myself a little bit bigger here just for a minute and talk to you. All right, now, bacteria, all right, can reproduce asexually. It's called binary fission. The single circular chromosome replicates and they just divide, okay? That's asexual reproduction, no variety. But when the conditions around bacteria start to change, it stresses them out. And they're like, you know what? Somebody might have some DNA that could help me out right now. And that's when they start copulating. That's when they start having sex. And they grow out of their cell. They grow this little sex pilus. It's like they're creating a little penis. And what happens is that sex pilus will extend to another cell and they can exchange DNA across that. That's how they can increase their variety and maybe get some other genes, all right? So that's called conjugation. Now, I want to tell you a third way bacteria can get variety of genes is called transduction, and that's when a virus that infects one cell grabs some DNA from host 1, and then that virus infects another cell and sends that DNA from host 1 into host 2. That's called transduction, all right? So that's what the sex pilus is all about. Okay, right here, tube used to pass DNA for reproduction. So sex pili, tubes used to pass DNA from cell to cell. You know, it's something kind of interesting. They can have like 3 of those going at one time. Okay, all right. Now, let's move. We did this, and this. Now we're going to move to the inside. Okay, so like I said before, the fluid portion, okay, usually outside of the nucleus or outside of the DNA region, the nucleoid region where this DNA is, that is the fluid of the interior of the cell is called cytosol. So cytosol contains water. Inorganic and organic nutrients, or inorganic and organic molecules, and enzymes. All right. The nucleoid region is that region where the single circular chromosome hangs out. So this is the nucleoid region. And when you look over here in this cell, this is the nucleoid region, kind of that lighter color in here. And that's that scrunched up, looks like a hairball kind of a chromosome in there. So region that contains the single circular DNA molecule. Single circular DNA molecule. Now, I have another picture for your next one, the plasmid. I think I need to actually skip 2. Oh, I don't. Okay, here it is. There we go. Okay, so this is a single cell, right? Here is the bacterial chromosome. You know that's not how it's going to be. It's all going to be much larger and crunched up, but they can have a secondary little tiny ring of DNA, and it's called a plasmid. And plasmids are still the genetic code. It's still genetic information. And what scientists will do, and in fact, if you were in my class this year in person, maybe you will be in the spring, we genetically engineer bacteria. And what we do is we take plasmids and we insert a gene for the color of like a mutated red color of like a jellyfish, and then we put that gene inside a plasmid and then get the cell to take the plasmid, and we transform the organism by that. And then they start turning this mutant red fluorescent color, just like the jellyfish, because it's the protein that they build. So genetic engineers like to manipulate plasmids. So on your notes, plasmids are small accessory rings of DNA that can be manipulated. Okay, now let me go back to where we were. Okay, so next one on there is ribosomes. And ribosomes are like the little dots that you see in there. And those are for protein synthesis. It's right here, protein synthesis. Okay, and now there's some other things on here you don't have to worry about. Inclusion bodies, they just store mesosomes. I want to point out to you just for a second here, this mesosome, it's increasing the surface area, right? So it has more abilities to absorb nutrients. Nucleoid region, we did. I think we got everything there. All right, now I am going to show you, this is archaea. Remember I told you there were 2 domains that are prokaryotic. Bacteria is one, archaea is the other one, and we'll come back to archaea in just a minute. All right, so here you can see all different kinds of bacteria all over the place. Yum. All right, I showed you the plasmid. Now, if I was going to pick a poster child of bacteria, I would pick Cyanobacteria, okay? Cyanobacteria, I would pick them for 2 reasons. One, they do a ton of photosynthesis for our planet, fixing CO2 into sugar. But remember from the cycles, the biogeochemical cycles that we talked about, and remember when we talked about the nitrogen cycle, and the first part, remember that the reservoir for nitrogen is in the air. So cyanobacteria do nitrogen fixation, where they convert N2 into ammonia N3, and then remember there's nitrification and all the rest of that. So on your notes, I have an example is cyanobacteria, and it is a major contributor to the oxygen revolution. That's about 2.3 billion years ago when oxygen filled our planet, and they still help today, and nitrogen fixation. All right, now let's talk about, you can take all of life and you can sort it into 3 baskets, right? All of life can be sorted into 3 baskets. Here are your 3 baskets: bacteria, archaea, and eukarya. All life is one of those 3. Now, archaea and bacteria up here, these are both prokaryotic. They both don't have any membrane enclosed organelles. They don't have a nucleus at all. Eukarya does have membrane enclosed organelles, and they can be broken down into these 4 different kingdoms. Now, what I want to point out to you, though, is differences between bacteria and archaea. And so on our notes, I've already talked to you so far. It's all been about bacteria. So now, what are the differences in archaea? So in archaea, their cell walls and cell membranes are a little bit different from bacteria. And their DNA and RNA structure is actually more similar to eukaryotes. So DNA and RNA structure is more similar to eukaryotes. And then many are found in really extreme conditions, like halophiles are where it's really salty. Methanogens live in cow rectums, producing methane gas. You have thermoacidophiles that live like in Yellowstone, where you see those pools of really, really hot, low pH, high temperature water. So archaea are known as extremophiles. Okay, so if you want to add that, they are extremophiles. extremophiles. All right. And then remember, just as a review, eukarya, you've got your 4 kingdoms. If I was going to talk about each one, I'd say fungi is multicellular. They are an absorptive heterotroph with walls made out of chitin. Animalia, fungi are mostly multicellular. Animalia are always multicellular, and they are ingestive heterotrophs. Plantae are photoautotrophs, doing photosynthesis with cell walls made out of cellulose. And Protista, he's kind of like the catch-all. Most of them are unicellular, though it just depends on what book you look at, how they include them. But most are unicellular, and they can be both autotrophs and heterotrophs. In fact, some act like plants, some act like animals, and some act like fungi. All right, and now just kind of as transition between this video and next, all right, when you look at prokaryotes versus eukaryotes, so right, this should look very familiar to you, a prokaryotic cell. And then in video 2, we're going to talk about eukaryotic cells. The only thing I do not like on this picture is the size reference. The scale and proportion is wrong because this prokaryotic cell would be about the size of this mitochondria. All right. So in conclusion, you should know the cell theory, right? The cell theory. You should know why cells are small. You should know the structures and functions of each of those structures of a prokaryotic cell. And you should know how archaea, also a type of prokaryotic cell, how it's different, how you differentiate it from the other prokaryotic domain, which is bacteria. Okay? And that's it. I hope you're having a good day.